Hello everyone, welcome to the new session of Biotechnology in Agriculture Part 2. In this session, we are going to study about the Bt cotton, right? And in the last session, we had covered about the RNA interference and delayed in fruit ripening. So let us start with this session. Bt cotton is genetically modified organism, GMO, that is genetically modified organism. So this is a cotton plant, that is the Bt cotton, and variety which produces an insecticide to bollworm. So this particular crop, that is the Bt crop, produces an insecticide that kills the bollworm. Strains of the bacterium Bacillus thuringiensis. So this is a particular bacteria, that is the Bacillus thuringiensis, and that initial BT, that is B for Bacillus and T for thuringiensis. Okay, so according to this particular bacteria, Bt cotton got its name. This variety of cotton got its name. Okay, so strains of the bacterium Bacillus thuringiensis produces over 200 different Bt toxins. So it produces 200 types of toxins, okay, and each harmful to the different insects. Okay, so most notably, that is the Bt toxin are the insecticidal to the larvae of moths and butterflies, beetles, cotton bollworms and guttu flies, but are harmless to other forms of life. So they are not harmful for all the forms of life. They are harmful for moths, butterflies, beetles, cotton bollworms, guttu flies. Okay, so these flies and these insects are very harmful for cotton plants. They are very harmful for the cotton plants. Okay, so the gene coding for the Bt toxin has been inserted into the cotton as a transgene. So Bt toxin, this particular gene, a particular gene from the Bacillus thuringiensis, this bacteria, is taken out. This gene is taken out and introduced to the cotton plant, and this cotton now got its name Bt cotton, right? Bt cotton. So we will see in the coming slide how this mechanism occurs. In many regions, the main pest in commercial cotton are Lepidopteran larvae. So this is a larvae that is called as the Lepidopteran larvae. Mostly they are infected to the commercial cotton, which are killed by the Bt protein in the genetically modified cotton they eat. Okay, so when they eat this particular variety of cotton leaves, they die off. So this eliminates the need to use large amount of broad spectrum insecticide to kill Lepidopteran pests. So we want to kill this particular type of insects or their pests so that our cotton plant be protected, right? This spares natural insect predator in the form of ecology and further contributes to the non-insecticide pest management. So this is the natural insect predator. This particular crop, that is the Bt cotton, is a natural insect predators. Okay, Bt cotton is ineffective against many cotton pests such as plant bugs, stink bugs and aphids. Okay, so they do not harm to these particular insects or the pests. Depending on the circumstances, it may be desirable to use insecticides. So they are not harmful to the plant bugs, stink bugs or the aphids. A protein that is toxic. Now we will talk about Bt toxin, a particular toxin that is produced by the Bacillus thuringiensis. A protein that is toxic to chewing insects and is produced by the soil bacterium Bacillus thuringiensis. So this particular bacteria is found where? It is found in the soil. Okay, and has a long been used as a biological pesticides. So we As a biotechnologist, what we do, we take out the particular gene that is responsible for producing Bt toxin and introduce to the cotton plant. And that in that way, the cotton plant produces that toxin. By means of genetic engineering, the genes for the Bt toxin can be isolated from the Bacillus thuringiensis and transferred to the plant, and that is particularly cotton plant. And that is why the plant got its name Bt cotton. So let us see how it happens. Bacillus thuringiensis is a bacteria that produces protein which are toxic to insects. So this particular bacteria produces a protein that is toxic to insects. This bacteria produces over 200 different types of Bt toxin, and each is harmful to the different insects. So this particular toxin is very harmful to the insects. The Bt toxin dissolve in the high pH insect gut and become active. So when this cotton leaves infected by the worms, okay? So what happens? They take that part of the leaves, and when it get in the intestine of this worm, what happened? The pH of insect gut is very high, okay, and that particular protein, that toxic protein, become active. Earlier it is in inactive form, but when the pH of the insect gut that is very high, that inactive protein become active. So this toxin then attack the gut cells of the insect, punching holes in the lining. So it punches the holes in the lining of the intestine. The Bt spore spills out the gut and germinate in the insect, causing death within a couple of days. So Bt spores are also there. Spores mean bacterial spores are also present in the this insect's gut. So that pores, what happened to that pores? Inside, through that pore, inside the bacteria which germinate from the pore get enter inside the body of the insect, and that causes death, okay? Even though the toxin does not kill the insect immediately, treated plants part will not be damaged because the insects stop feeding within the hours, okay? So what happened when this insect eat that particular part of the cotton leaf? What happened? Now it is stop eating. It is stop eating because its gut is infected with the bacteria now, and after an hour or within an hour, this particular insect dies off, okay? So the Bt spores do not spread to other insect or cause disease outbreaks on their own. So only the worm that eat cotton plant leaf, that only get infected. Others are not get infected, okay? Only the worm that eat the leaf of the cotton, that only get infected. So this is the step showing the formation of the Bt cotton plant. So what happened? Agrobacterium tumefaciens is taken, okay, and its plasmid is taken out. So this is a plasmid. Now we all know that what is a plasmid? That is, in the bacterial cell, there are some circular DNA apart from the main chromosomal DNA. Okay, this is a small circular DNA, and that is called as Ti plasmid. Ti plasmid. So this particular plasmid is taken out from the Agrobacterium tumefaciens. Okay, so this particular plasmid, that is called as Ti plasmid. Now it is having a tDNA, and that is the restriction site. Now we will take a restriction endonuclease and cut a portion of it. Now we will take out a DNA containing gene for the desired trait. Now what is the desired trait? In this case, desired trait is Bacillus thuringiensis bacteria we will take and take out that particular part of the DNA that is responsible for Bt toxin. Bt toxin. So that part is taken out. Here it is showing this part is taken, this particular red part. Okay, this is taken out from the Bacillus thuringiensis and it is inserted to the TI plasmid. So here we can see the recombinant TI plasmid. Why it is called recombinant TI plasmid? Because a gene of interest has been inserted into it. So now it become recombinant type and now it is known as recombinant TI plasmid. Now the next step, introduction into the plant cells in culture. So we will take out a plant cell and we will introduce this particular recombinant TI plasmid into that part. Okay. So what happened after the regeneration? What will happen? We will get a plant with a new trait. Okay. So with the help of tissue culture, we will generate a new plant. Okay. And this plant will have Bt toxin. Now, steps to show how the Bt cotton produces Bt toxin. Insect eat Bt crystals and the spores. Now, the leaf on which the insect is present, it is eaten by that particular insect. So that this particular leaf part is having that crystals, that is Bt crystals, and also the spores of bacteria. Now the toxin binds to the specific receptor in the gut and the insect stop eating. Now what happened? This particular toxin, is bind to the receptors that is present inside the lining of the gut. The crystal causes the gut wall to break down. Now this crystal causes gut wall to break down and allowing the spores and the normal gut bacteria to enter the body. So the gut wall is having some channel or the opening through which the bacterial spore get inside the cells of the intestine. And that means that bacteria get entered into the body of insect. The insect dies as spores and gut bacteria proliferate into the body. Now when the bacteria get enter inside the body, it proliferate. Proliferate means it divide and redivide and increases its number. So at this particular stage, what happens? Insect dies. Bacillus thuringiensis forms a protein crystals during a particular phase of their growth. These crystals contain a toxic insecticidal protein. The Bt toxin protein exists as an inactive protoxin, but once an insect ingests the inactive toxin, it becomes active. So what happened? The pH of the insect gut is very high. And in that case, inactive protoxin become active. It is converted into an active form of toxin due to the alkaline pH of the gut, which solubilize the crystals. So that is what we are talking about. So crystals, that toxin crystal gets solubilized. The activated toxin binds to the surface of the midgut epithelial cell and create a pores that causes cell swelling and lysis. Lysis means there occurs a channel or the opening inside the gut of the insect and eventually causes death of the insects. Specific Bt toxin genes were isolated from the Bacillus thuringiensis and incorporated into the several crop plants such as cotton. So this particular Bt toxin is not only introduced to the crop, that is the cotton, but it is incorporated to some other crops also like Bt brinjal. The choice of genes depends upon the crop and the targeted pest, as most Bt toxin are insect group specific. Now, as we all know that it produces 200 different types of toxin, and that is specific to the insects group. The choice of the genes depends upon the crop and the targeted pest. What is the target pest and what is the crop? So which crop is infected with the pest, that we should know. As most Bt toxin are insect group specific, so this Bt toxin is not a insect specific, whereas it is insect group specific. Means a group, a wide range of insect get infected with this Bt toxin. There are a number of them. For example, the protein encoded by the gene CRY. Now this is the protein that synthesizes, that is called as CRY protein. Now this protein is of different types. The genes that encode this protein are CRY1AC, CRY2AC and CRY1AB. So this particular two, CRY1AC and CRY2AB, control the cotton bollworm. So this particular two specific genes that control cotton bollworms, right? And this particular one, that is the CRY1AB, that controls corn borer. So this is very, very important from the examination point of view. The dissolved and the activated CRY molecule. So where this particular CRY protein get activated? Inside the gut of insect. Bound to the cadherin-like protein. Now, where this cadherin-like proteins? These are the receptors protein that are present on the cells of intestine of the insects. Okay, on cell comprising the brush border molecules. Okay, so the dissolved and activated CRY molecules bound to the cadherin-like protein on cell comprising the brush border molecule present on the intestine. Epithelial cells. In fact, the epithelium of the brush border membranes separates the body cavity from the gut whilst allowing access for nutrients. The CRY toxin molecule attach themselves to the specific location on the cadherin-like protein present on the epithelial cells of the MEG, and ion channels are formed which allow the flow of potassium. So this allow, now the ion channel, this particular ion channel forms and allow the flow of potassium. Regulation of potassium concentration is essential, and if left unchecked causes death of the cell. So this imbalance of the potassium concentration in the epithelial cell, okay, causes the death of the cells. Due to the formation of CRY ion channel. Now this particular channel, which is formed by the attachment of the CRY protein to the cadherin receptors, causes the formation of CRY ion channels. Okay, so due to the formation of this CRY ion channels, sufficient regulation of the potassium ion is lost and result in the death of the epithelial cells. So this potassium ion imbalance is caused because of the attachment of the CRY protein to the cadherin receptors, and that causes formation of ion channels. The death of such cells create gaps in the brush border membrane. So when these epithelial cell dies out, that creates a gap in the epithelial border cells, okay, brush border membrane. So here we can see this is the ingestion. Ingestion means the plant eat the part of the leaf, that is the BT cotton, and inside the intestine of this insect, what happened? High pH, due to the presence of high alkaline pH, what happened? This protoxin become activated and it becomes activated toxin. Now this activated toxin get bind to the receptor. Which receptor? Cadherin receptors, which are present at the membrane, membrane of the intestine, and that causes disruption of the potassium channel. And when this potassium channel disrupted, there is a death of the epithelial cell. Now this epithelial cell death causes pore formation. And what happened then? The spores, which are also present. Now here you can see this particular, these are the spores of bacteria. They germinate and get entered to the cell through this particular pore, which is caused by the death of the epithelial cell, and it proliferate there. Proliferate means divide and redivide and cause death of insect. BT action is very specific. Different strains of BT are specific to the different receptors in the insect gut wall. So the receptor which we talked about here in the case of BT cotton, that is cadherin, but this is very specific. So there are 200 different types of toxin, so they will have a different receptors in the intestine gut wall. BT toxicity depends on the recognizing receptors. So their toxicity is very dependent on the recognizing the receptor, and damage to the gut by the toxin occur upon binding to a receptor. Unless this toxin bind to the receptor, there will be no damage to the insect or the insect gut wall. Unless this BT toxin bind to the specific receptor. Okay, so this is very important step. Now each insect specific possesses different types of receptors that may match only certain toxin proteins. It is because of this that farmers have to be careful to the match the target pest. So we must know what are our target pest, okay, and with a particular BT toxin protein which is specific for that particular insect. So this also helps the beneficial insect because they will usually not be harmed. So there are many insects which is very beneficial for the farmers, right? They do not harm. In fact, they are beneficial in some or other respect. But if they get killed, then again there will be a great loss, right? So this is also help. Knowing the target is pest specific species is helpful the beneficial insects because they will usually not be harmed by the particular strains of BT. Okay, so let us talk about the advantages of the BT cotton. BT cotton has several advantages over the non-BT cotton. Increased yield of cotton due to the effective control of 3 types of bollworms. So this particular variety, that is the BT variety of the cotton, control the bollworms. What are those bollworms? American spotted and the pink bollworms. So it controls these types of bollworms. This particular variety controls these types of bollworms. Insect belong to the Lepidoptera, that is the bollworms, are sensitive to the crystalline endotoxic protein produced by the BT gene. So this BT gene produces a cry protein. Cry protein. We have seen those 3 different types of protein, right? Which in turn protect the cotton from the bollworms. Reduction in the pesticide use in the cultivation. So when there is a control of pest by using this particular variety of the cotton, there is no use of pesticide. Why to use this pesticide? Okay, so reduction in the pesticide use in the cultivation of the BT cotton, in which bollworms are the major pests. We can reduce the use of pesticides because they are easily get controlled by the use of this particular variety of cotton. Reduction in the cost of cultivation and lower farming risk. Okay, so this particular use of this particular cotton, that is causes the reduction in the cost of cultivation and lower the farming risk also. Reduction in the environmental pollution by the use of insecticide. Rarely, sometimes we use the insecticide and that causes pollution in some other way. Okay, so by using this particular variety of the cotton, that is BT cotton, causes the reduction in the environmental pollution also. BT cotton exhibit genetic resistance or inbuilt resistance, which is permanent type of the resistance and not affected by the environmental factors. So there are many environmental factors that do not affect this particular variety of the cotton. So that is good to use, right? So thus protects crop from the bollworms. BT cotton is eco-friendly and does not have adverse effect on the parasites, predators, beneficial insecticides and organism present in the soil. So this particular variety of cotton is not affecting to the insect that are beneficial to the farmers, right? So it is even not harming to the organism that is present in the soil. So this particular BT cotton is good to use. It promotes multiplication of parasites and the predators, which help in controlling the bollworms by feeding on the larvae and eggs of the bollworms. No health hazard due to the rare use of insecticide, and BT cotton are early in maturing as compared to the non-BT cotton. This is also one of the very important advantages over using the cotton type which is not of BT, right? So this is important. Now there are some of the disadvantages. High cost of BT cotton seeds. The seeds of the BT cotton are quite high cost as compared to the non-BT cotton seeds. Effectiveness up to 120 days. After that, the toxin producing efficiency of the BT gene drastically reduced. So it is effective to 120 days, and after that the effect of this gene get reduced. Okay. So ineffective against sucking pests like jassets, aphids and whiteflies. So there are some other insects that harm to the cotton, but this particular toxin is not going to control those species like jassets, aphids and whitefly. Okay. So this is all about this session, that is we have covered in this session about the BT cotton. This is one of the great advantages, okay, in the field of agriculture. Thank you very much.